All right, cell membrane part 1. The cell membrane is a very important organelle of the cell. It defines the cell, so it determines what is part of the cell and what is just the aqueous environment, the watery environment that the cell is in. The cell membrane is important because it can control what comes in and out of the cells. Some things can move through that membrane pretty easily, and some things cannot. All right, so this is a link that will be on the PowerPoint. You won't be able to access it on this video, but if you go to the PowerPoint, you can access it. So it's a pretty interesting video, and it looks at what microscopic organisms, what will happen if their cell membrane is broken. This is a phospholipid. Phospholipids are very important for the cell membrane. It makes up the bulk of the structure. Each one of these diagrams represents the same thing. These are all phospholipids. So this is how the phospholipid is written as far as its chemical formula. This is a space-filling model, so each one of these little dots represents an atom, and it shows how those atoms are bonded. And this is the icon. So this is normally what you're going to see because it's a lot easier and faster to write. But understand that all three of these, no matter how you see the model, it has two very important parts. The hydrophilic head. So hydrophilic, hydro means water, philic means loving. So the hydrophilic head on each of these molecules, it's attracted to water. So it's always going to be oriented toward the watery part. Hydrophobic, hydro meaning water, phobic meaning to fear or to try to avoid. So the hydrophobic tail on each of these molecules. So a phospholipid molecule has a hydrophilic portion to it and a hydrophobic portion to it. That's important because that helps it to arrange in the cell membrane. The cell membrane is currently, the fluid mosaic model is the current model for a cell membrane. And it is composed of a phospholipid bilayer. So bi means two, so two layers of phospholipids. So this is a very simplified version of it. So this was our little icon version with the hydrophilic head and the hydrophobic tails. And so notice whether it's outside of the cell or inside watery environment of the cell, the hydrophilic portions are always pointed toward the water. The hydrophobic portions are always pointed away from the water. Bilayer, I have two layers of phospholipids. Now the reason why they call it a fluid mosaic model is because of the fact that this is not like solid in its orientation. So the phospholipids that are within that membrane, they actually move around. In a membrane, there are weak hydrophobic interactions, and it can allow lateral movement. So it's going to allow movement from side to side. These guys just kind of switch places, or these two will switch places, or these two will switch places. The lateral movements can occur about 100 million times per second, but there's also a flip-flopping that can occur. So this phospholipid can switch places with that phospholipid. This flip-flopping is only going to occur maybe once per month. So the lateral movements are definitely a lot more common. The phospholipids, depending on how they are stacked together, can also give more or less fluidity to that membrane. We don't want a membrane to be solid. If a membrane is packed and is very solid, then it does not have a lot of flexibility, and it also is more susceptible to the environment, so it can break that membrane. And if you saw if you went back and watched that video clip of what happens to that one unicellular organelle when its membrane got broken, you know it's probably not a good thing for that to happen. Whenever fats are saturated, whenever lipids are saturated, they're going to be packed together really tightly. So these guys right here, these are saturated and they're packed together really tightly. These phospholipids have a hydrophobic tail that is unsaturated. It's got a double bond in there somewhere that causes it to bend. And notice how much more space there is between those phospholipids. They're not packed together as much. So these represent unsaturated fats. It increases the fluidity, and it doesn't get solid until a much lower temperature, so you have to cool it way, way, way down. There are benefits to this. So plants that have cell membranes that are unsaturated, they can survive lower temperatures. So if you or anybody in your family, if you're farmers, if you grow winter wheat has a lot more unsaturated phospholipids in the cell membranes. Cells that are more saturated, they can't take those lower temperatures. They pack together and they're a little bit more fragile. They don't have as much movement because they're not as fluid. Another component that can add to the fluidity of a membrane that has more saturated fats into it is adding cholesterol. So cholesterol gets a really bad rap. Everybody talks about cholesterol as being bad, but it's actually a very necessary to our survival, and it's a very key component to cell membranes. So adding this lipid in between our phospholipids, adding cholesterol in there, increases the fluidity and gives it more movement, which is a good thing. All right, so the cell membrane is not just those phospholipids packed together. There are a lot of other components that are within the cell membrane. Cytoplasm, this is the internal part of the cell. So this is going to be our external part of the cell. This is our extracellular fluid. These are filaments of cytoskeleton. Cytoskeleton helps the cell to keep its shape. We already know about the phospholipids, these little jellyfish-looking things with our hydrophilic heads and hydrophobic tails. Makes a phospholipid bilayer in our fluid mosaic membrane model. We already know about cholesterol. Cholesterol molecules that are embedded in that phospholipid bilayer helps to increase the fluidity, flexibility of our membrane. So these guys are considered transmembrane proteins. Trans, they go through it. Membrane, so our cell membrane, and they are proteins. They're made of amino acids, so they're a protein structure. This is a peripheral protein. Transmembrane proteins go through the membrane. Peripheral proteins, they're either going to be, within the inside or they're going to be on the outside, but they're not through the membrane. They're on the peripheral, they're on the side. This represents a glycoprotein because this is a protein right here, but this has this little guy sticking on top of it. And these are all sugars. Glucose is a type of sugar, so glycoprotein. All of this is bonded together and it's within the membrane, but it's a glycoprotein within the membrane. Glycolipid, it's got those lipid hydrophobic tail, but it's got a glucose on the top here, glucose molecules on the top here, so glycolipid. All right, transmembrane proteins. So there are so many different kinds of proteins that are so important to us. Once again, these all represent integral proteins, except for this guy right here is peripheral. It's not through the membrane. Some transmembrane proteins are transporters. So they basically just act as a channel. Some of these transporter proteins are open all the time. So they're like a door that's open all the time. Some of them have a gate on them, and that gate needs to be opened before anything can pass through. Some proteins provide a surface for enzymatic activity. So you have a molecule that bonds to the protein adjusts its shape in such a way that it either causes a bond to occur or breaks a bond. And so we'll learn more about enzymes later, but these proteins embedded within the cell membrane allow a place for enzymatic reaction to occur. Cell surface receptor. So this guy is embedded within the membrane, but it has a receptor on the surface, and a ligand protein can bond to that receptor, and it's going to cause a signal to go inside the cell. So this little molecule itself doesn't go in the cell. it just bonds with the integral protein, and then that protein transmits the message within the cell. Cell surface identity marker, we know these guys as antigens. So this is like the little name tag. If this were a red blood cell, then this might be a B antigen to identify that cell as a type B blood. Cell-cell adhesion. These are intracellular joining proteins, and they tend to be pretty long-lasting. So this would be a cell up here. This would be another cell down here, and these proteins bond to each other in order to keep those cells together in a tight formation in order to probably create a tissue. And then attachment to cytoskeleton. So this little peripheral protein is attached to the cytoskeleton to help that protein continue to keep its shape.